everybody. I'm Jillian Swinford. I'm a biologist with Massachusetts Division of Marine Fisheries. Today I'm going to talk to you about declines in Ovigerous female lobster in southern Mass. So everybody in this room know that commercial landings have declined sharply in this stock since the late 1990s, following a record high. Stock abundance indicators, as we just saw, consider the stock significantly depleted. And this is due to a variety of potential reasons, including there's a high shell disease prevalence in recent years, and then also increasing bottom water temperatures throughout the region. And because of this, fishery effort has declined, not only through trap reduction from fishery attrition, but also through policy as well. So once again, we are at record lows. So this, southern New England covers this wide swath of the Atlantic, but today we're going to be focusing on the inshore waters of Massachusetts, specifically Buzzards Bay, and then the waters just outside of Buzzards Bay and the Vineyard Sound. So before we talk about the question I have today, I'm going to talk about our two data collection surveys. We have commercial sea sampling and the eventless trap survey. So commercial sea sampling is fishery dependent. We're basically sampling lobster bins direct catch. It's been running since 1981. It targets large adults, so keepers, but also the eggers, oversized lobsters, V-notch females, anything that we throw overboard, we also get data on as well. And so in most years we target sampling during the fishing season, which is about May to November in this area. However, in recent years, we've had a harder time getting those late fall trips, and I'll kind of get into that a little bit later. For the ventless survey, it's fishery independent. It's designed by the state agencies. It's a random stratified sampling design. It's been running since 2006. In Massachusetts, there are 5 zones in the Gulf of Maine with 12 stations per zone, and then southern New England has been reduced to 1 zone with 16 stations. And for each station, there are 6 pot trawls with 3 vented traps and 3 non-vented traps. The non-vented traps retain anything that we catch. So this survey does target adolescent and undersized lobster. However, we do take biological information and data on anything that we catch. We sample from June to August in southern New England for this survey. So let's get into our main question here. So we do know that this stock has historically been declining, but the proportion of females with eggs in comparison to all eggers has remained somewhat consistent in comparison to each other, even with eggers increasing in that proportion. But recently we've seen this very sharp decrease right here in the proportion of females with eggs in comparison to females. And this is something we're also seeing in our ventless trap survey, which has a much shorter sampling period, as you can see by the blue box, but we're still seeing that same trend, and we want to know why are we seeing less eggers, essentially. So there's a couple things that could be preventing us from fully understanding this regarding our sampling methods. So we kind of want to address those things before we jump to worst case scenario here. So one, our sampling we could be sampling the wrong females, and the demographic pool has changed. We're sampling females at the wrong time of year, or worst case scenario, reproduction is failing. But we want to kind of check some of these boxes before we jump to that. So let's talk about our first question: has our sample pool changed? When we look at female length frequency, the range of sizes at which we are catching these individuals has not changed from year to year, although the size of the individuals has slightly increased. Our average size has slightly increased, and this is due to that gauge increase that happened around 2003. Which you can see a little bit more clearly here. We have that jump up in average size, but overall we are seeing average size of females and eggers increase annually, and this could be an indicator that the younger females aren't necessarily recruiting to the population in the same numbers as we've seen prior, and this could just be a response of the population just retaining those older, larger females. So has our size at maturity changed? Size at 50% maturity is 76.1 for this population. And essentially this is no change from when maturity was assessed in the 1980s in Southern Mass. And so we are effectively capturing that size at maturity within our sampling size range. Now the next question is, are these animals moving around in our sampling footprint or moving outside of our sampling footprint? Before we get into that, let's look at a bathymetry map of Buzzards Bay. Compared to the Gulf of Maine and other areas in Massachusetts where we catch lobster, it is fairly shallow. Our depth range here is like 0 to 20 meters, so not super deep. And because of that, it can get extremely warm in the summers in comparison to like Cape Cod Bay, for example. We do have two temperature loggers. We have that Cleveland ledge up north, and that's about 30-foot depth. And then we have Buzzards Bay barge down south, that's about 70-foot depth. And those are both monitoring bottom water temperature. So bottom temperatures above 20 degrees Celsius for lobster are generally considered suboptimal. One, because it can cause an increased disease prevalence, particularly shell disease, which just can increase physiological stress. We also see increased respiration stress in lobsters. And so this is kind of a bad threshold. So we looked at the cumulative degree days above 20 degrees Celsius and above at both of our loggers. We can see in our shallower logger, Cleveland Ledge, the blue, it's a lot warmer there than in our Buzzards Bay logger, but the trend is a lot steadier. In our Buzzards Bay logger, the deeper of the two, we start seeing an increase in the number of those degree days above 20 degrees Celsius. And so that is cause for alarm. We're seeing active warming, especially in those deeper areas. So the stock assessment also, agrees bottom water temperature has been worsening over time in this area. We're also seeing a higher ratio of female lobster outside of Buzzards Bay or in deeper water. And egg-bearing females are starting to shift into deep water as well. So our question is, are females and eggers retreating into deep water outside of Buzzards Bay as a thermal refuge and therefore outside of our sampling footprint? So I took a look at our data, running generalized additive models, looking at egger catch by depth of catch, which is D, and year, which is Y. And our partial dependence plots show us that we see an increased probability of egger catch at this 90 to 130 foot depth, which is about 27 to 40 meters. And we do see kind of a decrease in our commercial sea sampling once you get to those deeper depths, but I think that is just because we don't have that many data points for those deeper depths. So then we can take this data and look at if the females are moving more into that deep water. So this is telling us, this is the portion of total females in deep water out of all the females caught. And we see there's an increase in female catch in deeper water, that 90 to 130 foot depth over time. When we look at the eggers, we see a similar trend for commercial sea sampling, but not really a trend for ventless. There could be a couple things, reasons why this is. One, because we have a much shorter annual sampling period. It's only 3 months. And we also have a smaller sampling footprint. We're only operating in state waters for that survey. But what this means is potentially both eggers and females are moving into deep water. So when we add temperature into this with by using degree days, again, cumulative degree days, we see that the proportion of total females or eggers in deep water is increasing with the increase in the number of degree days. And so this may be telling us that as conditions are getting warmer, those individuals are moving again into those offshore, deeper water pockets. So this could be, confirming what we think we may be seeing in our data set. So our second question is, are we sampling during the right time of year? So egg stage is a really good way to know if we are hitting that reproductive season. So here we have the percent of females with eggs by month, grouped by egg stage. We also looked at this for the ventless survey, but again, we only sample 3 months for that. So we have a little less of a clear look with that. So what we're seeing here is we're capturing the sampling period pretty well, and we're also capturing it hitting about the same time of year for all of these different year bins. So our reproductive season hasn't changed. And what I did notice in this last year bin down here, so that's 2021 to 2024, we have kind of stopped sampling in that late fall period. And this could be partially contributing to why we're seeing kind of that decline of eggers in our own data set. Part of the reason is we just haven't been able to get trips to go out due to things like weather. Fishermen aren't necessarily going, or they're going like way offshore, and so logistically just does not work for us. So we determined that we're effectively capturing the reproductive period. But one of the other thoughts we had is if we are catching a higher proportion of females during a time of year where they don't have eggs, that could be skewing our data as well. So one of those periods is July to August, where we're seeing animals basically with no eggs because they've all hatched more or less at this point. But it's also the time when we're seeing some of the higher landings within this fishery. So this is the annual proportion of total female catch occurring only in July and August. And for ventless, we don't, again, really see a trend, but for commercial sea sampling, we see an increase in the proportion of females caught in July and August. So again, that could be adding to that sharp decline that we are seeing in our data. So the last question is the biggest scary one. Is reproduction failing? In our data, we see a decline in the females under 86 millimeters, so the undersized females in our ventless survey. And we've also seen in both our suction survey and the stock assessment survey, or the stock assessment, a decline in the young of year lobster density to the point we are not catching any young of year lobster anymore in our suction sites. So there is evidence for that younger group of animals declining in numbers. Now, our data set isn't really equipped to really answer this question. So we did turn to literature to see what was plausible. So one, are females reabsorbing vitelline from ova instead of spawning? So lobster can reabsorb vitelline. This has been shown to occur when the animal is stressed. And in this case, does stress equal those higher temperatures, those temperatures over 20 degrees Celsius? So this one is potentially a plausible reason why maybe we're seeing less eggers. So are females not receiving sperm from males, resulting in unfertilized eggs? So there is some research that indicates males pass sperm to females at a smaller volume when the females are severely diseased with shell disease. However, this is considered unlikely that shell disease is the main cause of recruitment issues in southern New England. So we determined this one to be unlikely. And then is fecundity decreasing? And yes, research has indicated that realized fecundity in current females is significantly reduced compared to historical females, and may be tied to increasing temperature as well. So this, again, is another potential reason why we may not be seeing as many eggers. So this whole presentation was an exercise in figuring out if this decline in the proportion of females with eggs is caused by sampling issues or by biological, environmental, or spatial issues. So let's go back over our questions. One, has our sample pool changed? So we determined that length, frequency, size range, and size at maturity we feel are effectively sampled and have not changed. So cross that one off. What we did see is an increase of female catch in deep water, and females and eggers are potentially moving out of that sampling range into deeper and cooler water. So that is plausible. are we sampling during the right time of year? So we determined that spawning cycle hasn't changed over time, but we are catching a higher proportion of females during summer when the presence of eggs is low, and so this could be a reason that we are seeing this. Is reproduction failing? Our data sets aren't currently set up to answer this question. There is evidence of declining small lobster in our data sets, including young of year, and is this occurring due to a lack of larval production? And literature indicates that potentially yes. So this is also plausible. Really quickly, go through some of our future research direction based on this project. So data analysis of relationships between female lobsters, eggers, and changing temperature in shallow and deep water at Buzzards Bay could help us understand the role that temperature plays in this, specifically looking at spatial relationships of catch with depth and size. Have lobster moved farther offshore? We can combine this potentially with other data sets in order to kind of fill in this puzzle piece. We could prioritize trying to get later fall sampling trips if possible. And then research into southern female lobster reproductive success could help us understand if reproductive failure is potentially the cause for this anomaly in our data. All right, thank you, and sorry for taking all the time. Try to go as fast as I can.
We do still have some time for questions.
Oh, really? Okay. I was running.
Thanks, Jillian. I just had a quick one, and that was if you are collecting data or at any point did collect data on like the relative clutch size in your ventless trap survey, so.
Yeah, no.
Okay.
We really, I mean, we do say whether it's a small clutch, an abnormally small clutch. We don't really have like any kind of scaling on that, so we could look into those that have like a small clutch comment, but beyond that, no.
Okay, thanks. Yeah, we've been collecting that and happy to share it. I'm not sure about the consistency over time.
Yes.
Qualitative, but.
Yeah. Okay.
Yes, great talk, lots of information.
Thank you.
I do sea sampling in New Brunswick too.
Okay.
And we've been noticing a lot of smaller clutches in quantity of eggs. So that's what we were wondering too. Not a decrease in eggers, but just a decrease in their amount of eggs. And so that's just a comment, not a question, I guess.
Yeah.
And also we've done tagging, and our females seem to be moving a lot further than the males.
Okay.
And they seem to be going more towards water that is less salty.
Okay.
So not necessarily temperature wise, although we do have temperature loggers, and that's also ongoing for research, but yeah. So maybe look into your river.
Yeah.
Do you have any rivers going?
I mean, we do, but not anything major that would like severely decrease salinity to a point that it would really be noticeable. Our main inflow and outflow through there is actually the Cape Cod Bay Channel. So you know, we do get some stuff flowing in and out Cape Cod Bay through there, but we don't really have a lot of fresh water input.
I'm definitely worried about egg quality.
Yes, yes.
But I'm interested to hear that you're just seeing less eggers in general.
Well, yeah, I mean, that's what we're seeing in the data, which is concerning, but I would ask actually Tracy, because she's been doing this a lot longer than I have we noticed an increase in the small clutch situation?
My gut wants to say yes.
Yeah.
The fecundity work that we published a couple years ago, Jason was the lead on that paper, and there's definitely a decrease in like so if you look at an 86 millimeter female, the clutch that she has now is way smaller than the clutch that she had back in the 80s when it was done that. So there's something going on. We haven't like just out the details or the causes yet.
Our sea sampling, it's an index of percentage when we do the clutch size. So it's like, is it 100% full or 75% full or 50 or 25? It's a 1 through 4
Interesting.
index.
Okay.
For clutch size, and it's almost never 100% anymore.
Interesting.
Yeah. It's
Okay.
really interesting.
That would be interesting to start including in some of our research, but yeah, we really only determine whether we think it's a small clutch or not.
If you could just respond briefly what Tracy said, just summarize that in about 10 seconds for the folks online without the mic.
So So, wait, I'm sorry.
Sorry, no, it's okay. I was just gonna, if you wanna just
Yeah,
pass the
we're
mic
good.
for Tracy
Yeah.
just for 1 second so that the folks online didn't miss out on the
Oh,
answers.
okay, I'm sorry.
Sorry.
Sorry, I didn't.
Yeah, sorry, I didn't wait for the mic.
And then
Yeah,
we'll,
the fecundity changes
wrap up.
we've seen, that the question was about smaller clutches. So my gut says yes, there's smaller clutches, we don't document that. We're actually talking about figuring out a way to standardize that technique in our surveys at the technical committee level, so we may be making a change there. Heads up, Jillian. But we have seen a decline in fecundity compared to historical. So Jason Goldstein was the lead on the paper, but like an 86 millimeter female now is carrying a much smaller clutch than an 86 millimeter female back in like the 80s when the work was originally done. So we have updated that. We've seen smaller clutches. And we don't have the solution for what's going on yet.
Great. Thank you very much.